Hello, everybody. Everybody's doing okay.
Can I just ask you a few lab questions before you start?
Sure, go ahead.
For lab 10 on part 2, the enzyme inhibition part, I'm a little confused about what the results actually indicate.
Okay. So just so that we're clear, for each part, you're using that color-coded chart that manual provides you. For part 2, you're looking at inhibition, and the key is to look at the components that are used to make up each of the test tubes. So test tube A has tyrosinase, catechol, and water. Test tube B, I believe, has tyrosinase, catechol, benzoic acid instead of water. And test tube C has tyrosinase, catechol, and resorcinol. So the idea, in a sense, is in your first test tube, you have the enzyme and you have the substrate, which you should have determined from part 1, that the tyrosinase sort of really reacts with. Now, what would happen if you sort of add a third component? In the test tube B, the third component is benzoic acid. In test tube C, I believe the third component is resorcinol. And the idea is, does inhibition occur? Now, you don't have to distinguish what type of inhibition it is, but does the benzoic acid and the resorcinol prevent the tyrosinase from working as it really should? That's the key idea in the part.
So then, since the benzoic acid, there was no color change, that means that it's not allowing it to work?
Yeah, exactly. So in other words,
right?
If the enzyme was really working, you would have a color of brown to the solution. But if you have no color change, essentially, then it means that the enzyme is not working. So you have a situation where, in theory, the tyrosinase should work with the catechol, but it's not. And so the question becomes, why? And in that case, the benzoic acid is the contributing factor to why.
Okay, thank you.
No problem. And then you sort of apply the same logic to tube C. Okay.
And then for question number 1 on the conclusion questions.
Sure.
It says, based on your results, how many substrates did the tyrosinase bind with in part 1?
Yep.
Do you know that based off the color change?
Yes. And if you think about enzymes being specific, how many of those different substrates should the tyrosinase work with, in terms of enzymes really showing a good deal of specificity? Just one.
Okay. So how would you know if it's working, if that's expected number?
Based on the color. So again, using that chart where negative is no color change, light yellow is plus, dark yellow is plus plus, and then brown is plus plus plus. The more pluses, the better the enzyme is working.
Okay. All right, thank you.
No problem. All right. Anything else before we get started? Again, the chat feature is open. Yeah, go ahead.
I also had a lab question for lab 11.
Yes.
The specific gravity results.
Yes, you don't have to do that.
How would we get that without, there's no video?
Yep, you don't have to do that part.
Oh, we don't have to do that part?
Nope, there's no video. We didn't make a video for that part. So you just have to do the other parts that are there. You don't worry about the specific gravity part.
Okay, so then would the one question get taken off the conclusion statement that he had to do with pH values?
Well, no,
I think it was how would you know if the pH value is normal?
So there is a video there where the pH of each of the urine samples are measured. It's more towards
Okay.
the beginning. It's probably, video 2 or 3 or 4 in that range. But the pH values of each of the urine samples are measured.
Okay, all right, I will look more into that. Thank you very much.
No problem. If you can't find it, just let me know, but the pH values for the urine samples were measured. All right, anything
That
else?
sounds good, thanks.
You got it. Anything else before we get started? All right, so the goal for tonight really is just to go through some problem solving with enzymes, which was sort of the second half of chapter 16, and then do some problem solving in chapter 17, dealing with nucleic acids and protein synthesis. So, let's just really just sort of jump into it. The examples that I'm going to be using for the enzyme part are coming from a different textbook. Unfortunately, I want to leave you the odd numbered problems in your textbook to sort of try as you get ready for the final. So I'm just going to pull these from, and I'll try to write out as much as I can for these questions. But I'm going to pull these from a different textbook. They're very similar but they are very similar to the questions. The session will probably go for 90 minutes, but again, I am recording it. So if you have to go at a certain point, it's fine. I'm going to post a video later on this evening. Okay. All right. So let's take a look at the first question. This is sort of match the terms. Match the terms to the correct statement. Okay, the first term is enzyme substrate complex. The second term is enzyme itself. And the third term would be substrate. All right. And then the first statement has a tertiary structure that recognizes the substrate. All right, so it has a tertiary structure that recognizes the substrate. Again, pretty straightforward types of questions. So we know the answer can't be substrate. What has a tertiary structure that recognizes the substrate? That, of course, is the enzyme. To be more specific, as you saw in the notes, within the large tertiary structure of the enzyme, there's often a small pocket whose shape really resembles that of the substrate. And that small pocket, of course, is what is known as the active site. But in terms of has a tertiary structure that recognizes the substrate, the answer to that would be enzyme. Letter B, the combination of an enzyme with the substrate okay, again, pretty straightforward, not too complex. The combination of an enzyme with the substrate. So we know that an enzyme will only work when the substrate becomes bound to the active site on the enzyme. And when the substrate is bound to the active site on the enzyme, you have what is known as an enzyme substrate complex. And keep in mind, it's only when you have this enzyme substrate complex that your reaction has an alternative pathway that has a lower activation energy associated with it. Remember, what do enzymes do? They make reactions go faster. How do they do that? They do that by lowering the activation energy for a reaction. The activation energy is the minimum amount of energy required for a reaction to occur. the less energy it takes for a reaction to occur, the faster a reaction can occur. And you only get this alternative pathway for a reaction that has lower energy when you have this enzyme substrate complex. What holds the substrate in the active site of the enzyme? A lot of the interactions that you have on sort of the tertiary level of protein structure, your hydrophobic interactions, which again are your dispersion forces, hydrogen bonding, and salt bridges as well. Those are really the main types of interactions that you have that hold the substrate into the active site on the enzyme. So keep all of that in mind. And then letter C has a structure that fits the active site of an enzyme. And we'll get into the ideas a little bit in a few minutes of how close, in shape the substrate and the enzyme have to be. But of course, what has a structure that fits the active site of the enzyme? That, of course, has to be the substrate. So one of the things you notice in the homework, and as you'll see as we go through our examples of enzymes this evening, it's a lot of sort of memorization of ideas and remembering those ideas to be able to answer questions. And then we'll see some examples of some one or two applications as well. So that was our first example. Let's do a similar one for our next example. So again, match each term to the correct statement. The first term in this case will be active site. The second term in this case will be induced fit model. And the third term in this case will be lock and key model. All right. So the first statement is an active site that has a rigid shape. Right. So basically, an active site that has a rigid shape, the answer cannot be active site. So what they're talking about here is sort of distinguishing between the induced fit model of enzyme substrate binding and the lock and key model of enzyme substrate binding. The model of enzyme substrate binding that talks about the active site having a rigid, sort of non-flexible shape is, of course, the lock and key model. Lock and key model describes the active site as having a rigid, non-flexible shape. So in order for the substrate to become bound to the active site on the enzyme, the substrate would really have to have an exact match in terms of shape of the active site. Just like in the analogy I like to use is like a door, to your house or something like that, where, there should really only be one key that goes into that lock, turns the lock, and opens it. Because inside of that cylinder, you have an exact match between the lock and the key that's used to open that door. So in essence, the lock and key model describes the active site as having a rigid, non-flexible shape, and only those substrates that have an exact, or only that substrate which would have an exact match for that shape of the active site would be able to bind to the active site and form the enzyme substrate complex. So letter A, lock and key model. Letter B, the portion of an enzyme where catalytic activity occurs. So just like we talked about in the last couple of slides, this slide and the last one, it's that active site on the enzyme where the substrate will bind to the enzyme, where we get the enzyme substrate complex, and you have now an alternative pathway for the reaction that has a lower activation energy. So pretty straightforward. And then letter C, an active site that adapts to the shape of the substrate. So again, now in this case, we're speaking to a model of enzyme substrate binding. And whereas the lock and key model describes the active site on the enzyme as having a rigid, non-flexible shape, the induced fit model, which is our answer to letter C, describes the active site as being able to adjust its shape, slightly adjust its shape to better fit or match that of the substrate. So essentially, in the induced fit model of enzyme substrate binding, there's sort of an interaction between the substrate and the active site on the enzyme, where essentially the active site on the enzyme can sort of adjust its shape slightly to better fit that of the substrate. Essentially, what you're doing is that you're inducing a fit. But one of the things that I always sort of point out is to keep in mind that wholesale changes in shape are not going to occur. Enzyme substrate binding is pretty specific. There are some enzymes that will really only react with one substrate. There are other enzymes that will react with a small group of substrates. But in reality, there's really not enzymes that react with a whole heck of a lot of substrates. So there really is a good deal of specificity in terms of enzyme substrate binding. But the shape of the substrate and the shape of the active site don't have to be an exact match. And the induced fit model of enzyme substrate binding is really the more accurate model of enzyme substrate binding, that there could be a little bit of interaction where the active site slightly adjusts its shape so that those interactions, dispersion forces, hydrophobic interactions, salt bridges, and hydrogen bonding, which would bind the substrate to the active site, could better occur. Okay, so I know this is very similar to one of the homework questions that was done, but I thought it was a good question to take a look at as well. All right, so let me just pull up another page to find some additional problems. All right. So one of the other aspects, sorry, it's okay. Yeah, so I know about the final exam. So what I'm going to do is I'm just going to post an announcement about the final exam probably tonight or tomorrow. At the end, I know if you have to go early, you won't be able to hear it, but I'm going to post some information about it tonight or tomorrow on Canvas. This way that everybody can see it. So I'll talk about that a little bit later on down the line, you know, at the end. So, but I'll post an announcement tonight on Canvas. And then if there's questions about it, you can let me know after I post that announcement. All right. Okay. So one of the other major aspects in terms of looking at enzymes deals with conditions that affect how well an enzyme works. And in the notes, we talked about a couple different things, and you saw it in the lab as well. We discussed it even a little bit late, a little bit at the beginning of our session tonight. And these are essentially factors that affect enzyme activity. And we talked about three different things. We talked about temperature. We talked about pH, and we talked about inhibition. So the next couple of questions that we'll take a look at with you will sort of deal with these aspects of it. All right. So here's our first example. Let's see. Okay. Suppose you have an enzyme that has an optimum temperature of 37 degrees Celsius and an optimum pH of 8.0. For each of the following changes, indicate whether the reaction rate will increase or decrease. Okay. So just to go back for a second, in terms of temperature, for each and every enzyme, there is a temperature where that enzyme is going to work at its best. That temperature is what is known as optimum temperature. For many enzymes, and not all of them, but for many of them, that optimum temperature is 37 degrees Celsius. What's the significance of that number? The significance of that number is that essentially corresponds to body temperature. So our bodies are set up to have most enzymes work very, very well. Now, usually under very cold temperatures, enzymes don't really work so well because the molecules of enzyme and substrate are not really moving. Remember, the whole idea in terms of temperature, in a sense, is based off the idea that molecules, even though you can't see them, are constantly in motion. In order for a substrate and enzyme, or for a substrate to bind to the active site on the enzyme, the substrate and enzyme molecules have to collide with each other. And upon collision, they have to recognize each other. And the general idea of recognition is that recognition is based on shape. If you're under very cold conditions, there's not a lot of heat, there's not a lot of energy present. So enzyme and substrate molecules are not really going to be moving. So even though you have enzyme present, there's not going to be a lot of interactions between enzyme and substrate. Now, as you start to warm things up, enzymes are going to work better and better, but there's going to be a certain point where that enzyme is going to work at its best in terms of temperature. That's the optimum temperature. And as you increase temperature from there, what starts to happen is that the enzyme actually starts to not work as well. So and how is this all reflected? It's all reflected in reaction rate. If the reaction rate, and that refers to how fast the reaction occurs, if the reaction rate increases, that's usually an indicator that the enzyme is working well. And if the reaction rate decreases, that's usually an indicator that the enzyme is not working so well. And what happens is that if you heat up an enzyme too much, what starts to happen is that you start to sort of impact the different interactions you have on the tertiary structure of the enzyme. And eventually, if you heat the enzyme too much, just like with proteins, what's going to happen to the enzyme? The enzyme's going to denature. For pH, it's a pretty similar thing. For each and every enzyme, there is a pH where that enzyme is going to work at its best. That is what is known as optimum pH. Because that pH best maintains the tertiary structure of the enzyme. It's the pH that best maintains the shape of the active site. And if you think about it, if that's the pH that best maintains the shape of the active site, as you go below or above, as you go away from the optimum pH, what happens to the enzyme? The enzyme's not going to work so well. You know, as you're going closer and closer to the optimum pH, the enzyme is going to work better and better. If you're going further away from the optimum pH, the enzyme's not going to work so well. And eventually, if the pH becomes too acidic or too basic, what could happen to the enzyme? The enzyme could denature. All right. So let's take a look at some situations. Okay. Changing the temperature of the reaction from 35 degrees Celsius to 20 degrees Celsius. So in other words, and again, you know, we're sort of thinking about the idea that, you know, you haven't, and let me just sort of add, for each of the following changes, indicate whether the reaction rate for a reaction catalyzed by this enzyme will decrease of this, for a reaction catalyzed by this enzyme will increase or decrease. I think you got to add that point in as well. Okay. Number one, changing the temperature of the reaction from 35 to 20 degrees, 35 degrees Celsius to 20 degrees Celsius. Okay. So you have a reaction that involves this enzyme where the reaction is running at 35 degrees Celsius. We take that reaction and we lower the temperature of that reaction from 35 to 20 degrees Celsius. Please excuse me for 1 second. So we change, so we have a reaction that involves this enzyme. And we have that reaction at 35 degrees Celsius, and we're changing the temperature down to 20 degrees Celsius. What's going to happen to the rate of the reaction? So at 35 degrees Celsius, we're running this reaction at a temperature that's close to the optimum temperature of the enzyme. We're taking that and we're lowering it down to 20 degrees Celsius. So what are we doing? We're moving the temperature further away from what the optimum temperature of the enzyme is. And if we're moving the temperature away from the optimum temperature of what the enzyme is, what should happen to the rate? The rate should decrease. So the rate decreases. Okay, so that's number one. Number two, changing the temperature from 45 degrees Celsius to 40 degrees Celsius. Okay, so now we have the sort of same reaction where we have the temperature of the reaction at 45 degrees Celsius, and we're going to lower the temperature again. We're going to lower the temperature from 45 degrees Celsius to 40 degrees Celsius. At 45 degrees Celsius, we are further away from the optimum temperature as compared to 40 degrees Celsius. So from going from 45 degrees Celsius to 40 degrees Celsius, we're actually, by lowering the temperature 5 degrees, we're actually moving closer to the optimum temperature of the enzyme. And because we're taking the temperature and we're moving it closer to the optimum temperature of the enzyme, what should happen to the rate? The rate should actually increase. Now, one of the assumptions that we're making here is that at 45 degrees Celsius, the enzyme hasn't denatured. That's probably a pretty safe assumption. It wouldn't, so if we said lower it from, if we said lower it from like 70 or 80 to 40, that would sort of be like a trick question in a sense, because if that reaction was probably at 70 or 80 degrees Celsius, there's a good chance that the enzyme would be denatured. And even if we lowered the temperature back down to the optimum temperature, the enzyme's not going to work. So maybe the one caveat that we'll put here is that you could assume the enzyme is not denatured at 45 degrees Celsius. So we can sort of add that caveat as well, just so you understand that if the enzyme was denatured, if we're at 70 or 80 degrees Celsius and we lowered it all the way back to even optimum temperature, the enzyme's not going to work. The enzyme's not going to work. So we want to keep that in mind. So that's number 2. Number 3, changing the pH from 6.0 to 7.0. So we're changing the pH from 6.0 to 7.0. So here we start at a pH that's further away from the optimum pH, which was 8, and we're changing it to a pH that's closer to the optimum pH. So what should happen to the rate? The rate should increase. And then the last one, number 4, changing the pH from 9.0 to 10.0. So here, just like in number 3, we're raising the pH, but this time we're in the raise of pH, we're actually moving further away from the optimum pH. And because we're moving further away from the optimum pH, what should happen to the rate? The rate should actually decrease. All right. So again, the idea in terms of optimum temperature, optimum pH, one of the things to keep in mind is that as you sort of get closer to it, the rate should increase because the enzyme should work better. As you get further away from it, the rate should decrease because the enzyme should not work as well. All right. All right. Let's take a look at one more example, and then we'll switch to nucleic acids. All right. So again, what we got? Match the type of inhibitor to the following statements. And remember, the three types of inhibition are competitive, non-competitive, and irreversible. All right, as we go through the different statements, that will allow us to talk about the differences in terms of the types of inhibition. The similarity between all of them is that when you have an inhibitor that's bound to the enzyme, whether it's in the active site or not, when the inhibitor is bound to the enzyme, the substrate cannot bind to the active site on the enzyme. And therefore, the enzyme is not going to be able to catalyze the reaction. All right. Letter A forms a covalent bond with an amino acid side chain group in the active site. This type of inhibition, when you have an inhibitor that's actually able to form a covalent bond, like a single bond, with a side chain group of an amino acid in the active site, this type of inhibition is irreversible. Because covalent bonds, especially single bonds, are not easily broken. So when your inhibitor is actually able to form a covalent bond with the side chain group in the active site, that basically means the inhibitor is going to permanently occupy the active site. And therefore, this type of inhibition is what is known as irreversible inhibition. Letter B has a structure similar to the substrate. What type of inhibition does this refer to? This type of inhibition refers to competitive inhibition. Remember, competitive inhibition is a race between the substrate and the inhibitor for the active site. Because their structures are so similar that the active site on the enzyme recognizes both. And if the inhibitor is in the active site on the enzyme, the substrate cannot occupy the active site on the enzyme. So this statement refers to competitive inhibition. All right. Letter C. Bonds to the surface of the enzyme causing a change in shape of the enzyme and subsequently the active site. And what type of inhibition is this statement referring to? This statement is referring to non-competitive inhibition. So remember, in non-competitive inhibition, the shape of the inhibitor and the shape of the substrate are not similar to each other. So there's not a competition for the active site. But what a non-competitive inhibitor does is that it bonds to another area of the enzyme. And when it is bound to the enzyme, the three-dimensional shape of the enzyme becomes distorted. And therefore, the shape of the active site becomes distorted. And if the shape of the active site is going to become distorted, the substrate is not going to be bound to the active site on the enzyme, or the substrate cannot become bound to the active site on the enzyme. So that's letter C. Letter D. The addition of more substrate does not reverse this type of inhibition, but the removal of the inhibitor by chemical reagent can return activity to the enzyme. And what type of inhibition is this referring to? This, of course, is referring to non-competitive inhibition. Since the shape of a non-competitive inhibitor and substrate are not similar to each other, you could add more substrate all day long. If the non-competitive inhibitor is bound to the enzyme, the shape of the enzyme is still going to be distorted. However, a non-competitive inhibitor can be removed from the enzyme by a certain chemical reagent or two, or maybe more. But it's not permanent because you can get the inhibitor to come off. And when the inhibitor comes off, the shape of the enzyme is going to return to what it should be. And therefore, you have restoration of the shape of the active site. So you return activity to the enzyme. Non-competitive inhibition. And then the last one, the addition of more substrate reverses this type of inhibition. And of course, this statement refers to competitive inhibition, because since it's a race between the inhibitor and the substrate for the active site on the enzyme, if we add more substrate, we could tilt the odds in favor of the substrate getting to the active site first. And if we tilt the odds in favor of the substrate getting to the active site first, that's going to slow or reverse the inhibition. So in competitive inhibition, if we add more substrate, that can really reverse the type of inhibition. All right. So that's a little bit, some practice problems involving enzymes. Okay. So in our final topic, we take a look at a little bit of nucleic acid and protein synthesis. So let's start with nucleic acids first. And for our nucleic acids, we know that they are made up of your nucleotides. Just in a sense, like proteins are made up of amino acids, your nucleic acids are made up of nucleotides. And let's start with nucleotides first. nucleotides essentially have three components. You have a nitrogenous base, you have a 5-carbon sugar, and you have a phosphate group. Now, you don't have to memorize the structures of the nitrogenous bases and the 5-carbon sugars. So I'm not going to say draw adenine or draw thymine or draw ribose or such like that. However, with them, there are certain aspects that you need to remember. And you'll see this in some of the problem solving that that we'll do with you in the next 45 minutes or so. But again, you don't have to memorize the structures of the nitrogenous bases. You don't have to memorize the structures of the 5-carbon sugars, but there are certain aspects of them that you should really become familiar with. All right. So let's take a look at our first example. State whether each of the following would be found in RNA, DNA, or both. And remember, in terms of nucleic acids, there are two types of nucleic acids. There's ribonucleic acid and there's deoxyribonucleic acid. Ribonucleic acid, of course, referred to as RNA, deoxyribonucleic acid, known as DNA. And one of the things that we'll go through as part of our discussions are the similarities and the differences between them. All right. Number one. And let's see if we can write a 1 that looks like a 1. All right. So first one, let's do guanine. So guanine, of course, is what? Guanine is a nitrogenous base. Remember, RNA and DNA have 4 nitrogenous bases. 3 of them are common to both. Is guanine one of those nitrogenous bases that are common to both? And the answer is yes. So guanine would be found in both. So that's number 1. Number 2. Thymine. Now, thymine is also a nitrogenous base, but it's actually one of those nitrogenous bases that's not common to both. So, in which nucleic acid in the nucleotides that are found in them could you find thymine? Would they be found in nucleotides found in RNA or nucleotides found in DNA? Thymine is a potential nitrogenous base that would be found in DNA.
Is there a trick to know this or just memorization?
Yeah, just really got to memorize it. You really just have to know that adenine, cytosine, and guanine are common to both. And then thymine is the 4th nitrogenous base in DNA. And, uracil is the 4th nitrogenous base in, RNA. If you want to sort of remember RU, so like RNA uracil, you could try to do it that way, but really that just comes down to memorization.
Okay, thank you.
You're welcome. Number 3. Adenosine monophosphate. So adenosine monophosphate is actually the name of a nucleotide. You saw in the chapter a little bit about naming, where for the name of a nucleotide, you take the name of the nucleoside and you add the ending monophosphate, or how it was taught at one point, 5' monophosphate. Now, one of the tricks to solving this one is, yes, adenine, because adenosine refers to which nitrogenous base? Adenosine refers to adenine. And remember, adenine is one of those bases that's common to both RNA and DNA. But the trick here is that adenosine monophosphate, the nucleoside name adenosine identifies it as being in what? It identifies it as being in RNA. Because remember, if you think about the nucleoside name, the difference between what you would have in DNA versus RNA is that in DNA, in front of the nucleoside name, you would have the prefix deoxy. When you don't have the prefix deoxy in front of the nucleoside name, then it refers to something in RNA. So if you had deoxyadenosine monophosphate, the answer would be DNA. But since here you only have adenosine monophosphate, that refers to RNA. Not both, not DNA, RNA here for number 3. And let's do one more, number 4. Let's do deoxy guanosine monophosphate. Now again, this is another name of a nucleotide. And if we think about the nitrogenous base, guanosine, guanosine refers to which nitrogenous base? Guanosine refers to guanine. But here, if we focus on the nucleoside portion of the nucleotide name, we have the term deoxyguanosine. So in front of the guanosine, we have the prefix deoxy, which is the indicator that this nucleotide would only be found in DNA. Again, if you had guanosine monophosphate, that would be found in RNA. But if you have deoxyguanosine monophosphate, that would be found in DNA. So, in terms of the nucleoside names, and again, you don't have to memorize the nucleoside names. But if I gave you something like adenosine monophosphate and asked you if it's an RNA, DNA, or both, you should be able to tell me RNA. If we give you deoxyguanosine monophosphate and ask you if it's an RNA, DNA, or both, you should be able to tell me DNA. Okay. So keep that in mind. So that's our first example. All right. Second, draw a nucleotide made from the following components. Let's see. Here's our first component. Actually, you know what? Change it up a little bit. Sorry about that. Uh, Second component. And then here's our third component. So I can just, all right, so what do we got here? We've got base, we've got sugar, we've got phosphate. So the three components of a nucleotide are, of course, phosphate, sugar, and base. And if we give you the three components of a nucleotide, we need you to be able to put them together in order to give us our nucleotide. So, a couple things. First off, in terms of the nitrogenous bases, you need to know which nitrogens are going to be the ones that are going to be linked up to the sugars. And if you go back into the notes, we circled the hydrogen on the nitrogen for each of the nitrogenous bases. That would be removed when the base becomes connected to the sugar. Again, you don't have to memorize or know what the name of this nitrogenous base is, but if you look at its structure, you need to remember that the nitrogen that's going to become connected to the sugar is this nitrogen right here. And it's going to be this hydrogen here that's going to be removed. So again, when you go back and look at the notes involving the different nitrogenous bases, you need to know which nitrogens are going to become connected to the sugars and which hydrogens are going to be removed in order for that to occur. Now, let's go to the sugar. If we're making a nucleotide, there are two important carbons that we want to remember. And that's carbon 1' and that's carbon 5'. And remember, the carbons in the sugars are numbered with primes to distinguish them from the carbons in the nitrogenous bases. We know that at carbon 1', you're going to have the link to the nitrogenous base. And at carbon 5', you're going to have the link to the phosphate group. Essentially, each of those linkages involve removing water. Because essentially off of carbon 1', I'm going to take off the OH, and I probably should have circled that. So the OH and the H, that's going to be one water that's going to be removed. And then for the phosphoric acid, we're going to take off the OH, and off carbon 5', we're going to take the hydrogen, and let me just see if I can write on 5' a little bit better. Sorry. off of carbon 5 prime, we're going to take off the H. So essentially those, and I should have squared that instead of circling it, of course. Those are going to be the waters that are going to be removed. Okay. So going from left to right, we're going to do phosphate, sugar, base. All right. So we'll start with phosphate first. And the part that's going to be removed is the OH off of the phosphate group. That's going to become, the phosphorus is going to be connected to the oxygen on carbon 5 prime. So the phosphorus becomes connected to the oxygen on carbon 5 prime. To give you what type of functionality, to give you what is known as a phosphate ester. P double bond O, O, C, phosphate ester. Then I'll fill in the rest of the sugar. Give myself a little more. Now, once I get to carbon 1 prime of the sugar, when the OH comes off and the H comes off the nitrogenous base, I'm going to connect carbon 1 prime to the nitrogen, the correct nitrogen on the nitrogenous base. And then I'm going to have the rest of the structure of the nitrogenous base. And the connectivity between the sugar and the nitrogenous base is another example, in a sense, of a glycoside bond. It's different from what we saw for carbohydrates. For carbohydrates, we saw ether type functionalities. But here in general, we still have another example of a glycoside bond. Okay. And this would be the nucleotide. Phosphate, correctly connected to sugar, correctly connected to base. Now, one of the other questions that's usually asked is that, is this nucleotide found in RNA or DNA? Now, there's a couple ways to answer this question. One way would be if you recognize which nitrogenous base we're using here. I'm not telling you which one it is. And again, you don't have to memorize the nitrogenous bases, so you might not know exactly which nitrogenous base it is. So then the question becomes, how do you identify it? And what you have to remember to do this is that the difference between ribose and deoxyribose is in the downwards direction off of carbon 2 prime. For ribose, what goes in the downwards direction off of carbon 2 prime? OH. For deoxyribose, what goes in the downwards direction of carbon 2 prime? H. So since we have an OH going downwards off of carbon 2 prime here, it indicates that this nucleotide would be found in RNA. The nitrogenous base that I used here was uracil. And remember, uracil is only found in RNA. So if you recognized it as uracil, you would know that this would be found in RNA. But really, for the way I sort of remember it, is just keep in mind the difference between ribose and deoxyribose, which would be at carbon 2 prime. Right? At carbon 2 prime.
So for the test, do we have to draw this out or just be able to pick things off of it?
No, you should be able to draw it out. If I give you the sugar, the nitrogenous base, and the phosphate group, you should be able to put them together to come up with a nucleotide.
Okay, is this gonna be a big part of the test or just a small section?
So it would just be like one question on the final exam to do this.
Okay, thank you.
No problem. Yeah, but this will sort of just be one question. And again, I'll talk more about that when I post my announcement later on this evening. All right. Just to also mention, because I think it's important, when you so this is how you put nucleotides together, or how you put a nucleotide together. One nucleotide, sorry. When you link nucleotides together to give a nucleic acid, remember the connectivity that you end up having really connects the 3' carbon of the sugar in one nucleotide to the 5' carbon of the sugar in the next nucleotide. And as we saw in the notes, and I'll just sort of circle it here, the connectivity that you have that links nucleotides together is an example of what is known as a phosphodiester bond. Where do we see phosphodiester bonds previously? We saw phosphodiester bonds previously when we talked about glycerophospholipids and sphingomyelin. So here, for phosphodiester bond, you have P double bond O, OC once, and then you have P double bond O, OC twice. That's your phosphodiester bond. And you have this when you link nucleotides together. We talked about when you're putting a nucleotide together, the phosphate connects to the sugar with a phosphate ester. The sugar connects to the nitrogenous base with a glycoside type bond. But here, when you link nucleotides together, you get a phosphodiester bond. Now, the difference between nucleotides in a nucleic acid is the nitrogenous base that is present. Because think about it, if you are in DNA, for each and every nucleotide, the sugar's got to be the same, deoxyribose. Phosphate is always the same. What's the only variable from one nucleotide to the next? The only variable is the nitrogenous base, because there's 4 different ones. If we're talking about DNA, there's cytosine, guanine, thymine, and adenine. That's the variable. As you go from one nucleotide in a nucleic acid to the other, the variable is the nitrogenous base that's present. So keep that in mind as well. All right. So let's talk a little bit more about DNA And in our next example, let's write the complementary base sequence for the following DNA segment. So remember, the structure of DNA is essentially a double helix. Two what are known as polynucleotide strands, in other words, many nucleotides linked together, winding about each other like a spiral staircase, where on the outside of the staircase, you have the sugar and the phosphate group, so they're like the railings of the staircase. And then you have the nitrogenous bases sort of arranged as steps on the inside. And what holds the two strands together in the double helix is hydrogen bonding between nitrogenous bases. And one of the key points is that the hydrogen bonding between nitrogenous bases is very specific. We talked about, or you saw in the notes, that adenine only hydrogen bonds with thymine, and cytosine only hydrogen bonds with guanine. And essentially, these two combinations, adenine with thymine, cytosine with guanine, are what are known as complementary base pairs. Now, you don't have to identify the hydrogen bonding in each. You know, what you'd want to remember is that for adenine and thymine, you have two hydrogen bonds between them. We're not going to ask you to sort of draw it out, but you just want to remember that there's two hydrogen bonds between them. And then for cytosine and guanine, there are three hydrogen bonds between them. But again, the hydrogen bonding is very specific. Adenine and thymine will only hydrogen bond with each other. Cytosine and guanine will only hydrogen bond with each other. So if you had the following DNA segment, and we'll use the one-letter abbreviations, ATA, GCCC, TTA, CTGG, got it all. What would be the complementary base sequence? So if you had adenine, so we'll do the original on the top, the complement on the bottom. So if the original has adenine, your complement is going to have thymine. If your original has thymine, your complement is going to have adenine. Then we come across adenine again, which would give us thymine. We have guanine, which gives us cytosine. Then we have three consecutive cytosines, which would give us guanine, guanine, guanine. Then we have two thymines, which would give us two more adenines. Then we have adenine, which would give us thymine, cytosine, which would give us guanine, thymine, which would give us again adenine, and then guanine and guanine, which would give us cytosine and cytosine. Okay, so one of the things that you do have to remember is you do have to remember these complementary base pairs. As you go from one DNA segment, or as you have these complementary, you know, if you have in the DNA double helix, you have two complementary DNA strands, which are held together by hydrogen bonding between complementary bases. So keep that in mind. Your DNA double helix, two complementary DNA strands winding about each other like a spiral staircase, held together by hydrogen bonding between complementary bases. All right. The other thing I wanted to mention in terms of DNA, I just wanted to mention a little bit about DNA replication. So keep in mind with DNA replication is that the two strands in the original, or what is known as the parent DNA molecule, separate. Enzymes are going to break the hydrogen bonds that exist between the complementary bases. But here's sort of the key idea. The two separated strands don't make copies of themselves. They make copies of their complementary strand. But since the two strands are complements in the original or parent DNA molecule, since the two strands are complements of each other, even though they're not making copies of themselves, even though they're making copies of their complementary strand, both original strands end up being copied. The two original strands just don't make copies of themselves. The two original strands make copies of their complementary strand. And what you get is you get two new DNA molecules identical to each other, and the parent DNA molecule called daughter DNA. Where each has one original strand and one new strand. So every time a DNA molecule gets replicated, you don't get one molecule, you actually get two DNA molecules. These two new molecules are what are called daughter DNA. The two daughter DNA are not only identical to each other, but they're also identical to the original or the parent DNA. And the daughter DNA just like the parent has two strands, one original and one new strand. The copy of the complement, the new strand, is the copy of the complement it made. So the original strand makes a new complement, and then they become hydrogen bonded to each other. To give it, it's not as simple as that, of course, in a sense, but that's essentially what occurs in terms of DNA replication. But one of the mistakes students commonly make is they think that the original strands make copies of themselves. No, the original strands make copies of their complementary strands. All right. All right. I said I was going to go for an hour and a half, so we're almost actually out of time. I wanted to do a couple of examples, talk a little bit about RNA, but I don't want to rush through it. And I know I was asked to talk a little bit about the final, so I'll mention that as well before we say goodbye. So what I'll do is a little bit later on this evening, I'll just do a short Zoom on my own that has some questions involving RNA and such like that, and then I'll post that to Canvas. And then if you have questions about that, you can email me and I'll get back to you that on as well. So before I say goodbye, let me just talk a little bit about the final. So as it says in the course syllabus, the final exam is cumulative. It's going to have two parts. Part one is going to be multiple choice, and that's really going to be the cumulative part. So it'll be sort of spaced out evenly. I can't tell you how many questions exactly, but it'll be spaced out evenly in the sense of the entire course, starting from acids and bases, and then all the way until what we did in the nucleic acid and protein synthesis chapter. So that'll essentially be part one. That will make up essentially 80% of the final exam. The other part of the final exam, part two, will be similar to the lecture exams, covering just enzymes and chapter 17, nucleic acids and protein synthesis. So those questions will be similar to the part two that you have on the usual exams, but those questions will only cover enzymes and nucleic acids / protein synthesis. Part two will make up 30% of the final exam. So you might say 80% and 30%, that doesn't add up to 100. It adds up to 110. What I usually do for the final exam is students can score up to 110 on the final. So that's your chance to really sort of boost up your grade if you need a little bit of sort of boosting up. Okay, so that's sort of the outline of the final. Part one, 80% multiple choice. Don't know how many questions yet. That'll be the cumulative part. Part two, sort of short answer, similar to part two of the lecture exams, covering just enzymes and then nucleic acids and protein synthesis. Okay. No, there's no curve. There's no curve. We go by the college grading scale. So whatever your grade turns out to be, based on the percentages in the course syllabus, that's what your grade is for the course. I'm not aware of curving that goes on at the college. There might be some, but I'm not aware. Other questions? All right. So as I said, chapter 16 homework, some of you sent, I already gave it back. Chapter 17 homework, if you want that back before the final, just get that to me as soon as possible. As I said, later on tonight, I'll do a short Zoom going over some RNA questions. And then I'll post that to Canvas as well. And I'll post the video for this session also. All right. Any other questions? Yes, go ahead.
Real quick, sorry. When will we have all the labs graded so we can kind of judge where we're at for this class?
So at this point, you have, I believe, 4 or 5 labs that are back. I don't think I'll have all the labs back to you before the final exam, but I think, after 5 labs, you have sort of an idea of where you stand in the lab portion of the course. The reality is that the lecture exams make up 45% of the final grade. So, in most instances, and I know the C+ is really the main concern for most students, in order to really get the C+, your average on the exams really has to be somewhere, no lower than high 60s, low 70s in order to get up a C+ for the class. But I think with 5 labs back, you have a pretty good idea of where your labs stand in the class. So, you know, you could really sort of estimate what you need to get on the final in order to get the C+ for the class. If you'd like me to do the calculation itself, you know, you could just email me to ask to do the calculation, and I'll give you an estimate of where you stand on, need to get on the final to get up to that C+ for the class. Other questions? All right. Thank you for taking the time to join this evening. Hope everybody stays well, and we'll talk to everybody soon. All right. Take care, everybody.